Hi everyone, I'm Dr. Anusha and today we are going to learn a very brief but important topic of microbiology, which is toxoplasmosis. So let's go straight into the topic. So toxoplasmosis, we can break down the term toxo, which means that it is toxic to pregnant females. So we can imagine that it is going to cause some disease in the fetus. Okay, so it is said that pregnant women should avoid cats. So we know that cat is of some importance here. We can also see that the mascot of this topic is a cat. Now, if you look closely to this cat, this has a big head and it has a spotted head. It has a big spotted head and red eyes. Why are these important? We will see later, but we can just remember that there is a cat with a big head, spotted head, and red eyes. Now, also from cat, we can remember CAT scan or CT scan. Why is the CT scan important? We will see as we progress. So we can look at the classification. Toxoplasma belongs to the kingdom protozoa. So protozoa are the unicellular parasites. Unicellular parasites also called as protista. It belongs to the phylum Sporozoa. These Sporozoa have spores. Okay, they are unicellular protistans which have spores inside them. It belongs to the class Coccidia. Now the organisms belonging to the class Coccidia are also called as epicomplexa or epicomplexan parasites. Now these are epicomplexa because they have an apical complex. What does this apical complex do? This helps in invading the cells. So it makes them into intracellular parasites. Now toxoplasmosis only invades the nucleated cells. So it will not be found in RBCs. So this differentiates your protestant toxoplasma from plasmodium, from malaria causing parasite, because it does not infect the RBCs. It only infects the nucleated cells. Let's look at the order. It is the Eumeridia. So gets our organism Toxoplasma gondii. Now we can remember gondii. It's derived from the origin. So most of the parasites, their names are derived either from the geographical location where they originated, the host that they infect, or any other characteristics. It may be size and shape. So we can look at gondii. It is derived from the name of the rodent, a small rodent from whom this parasite was discovered by Nicole and Montjoux in 1908 in Tunisia, North Africa. And Toxoplasma, the word toxo, the part, is derived from Greek letter or word toxon. Toxon means bow, the bow of bow and arrow. So the bow and arrow bow is the crescent shape of the trophozoites. What are these trophozoites of Toxoplasma called as? They are called as tachyzoids. We will look more into details of these tachyzoids. For now, we just have to remember the shape of the tachyzoid. It is a bow or crescent shaped with a broader posterior end and a narrower anterior end and a nucleus inside. Okay. Going on to the morphology, this organism mainly exists in three main forms. Okay. Three main forms: tachyzoids, tissue cysts, and oocyst. Okay, what is important to know that in all of these three forms, this organism is infective to man. We know that it is any one or a specific form of any parasite which is infective to man. But in toxoplasmosis, all three forms, all three stages of the parasite are infective to man. Let's look at the tachyzoid, our first form. So we understand, what do we understand by tachyzoid? So tachy stands for fast. So they are fast multiplying forms. Okay, these are the trophozoid forms which multiply very fast. So you can look at this Jeansa stained picture. Okay, and you can see these bow-shaped trophozoids, right? These are the tachyzoids. Now, the tissue cyst form, this, as the name suggests, it is found inside the tissue. So wherever the organism resides inside the body, in whichever tissue, these stages will be seen. So you can see that there is a cyst wall, and inside the cyst you have the trophozoid stage. Now at this point, this trophozoid stage is called as bradyzoids. Bradyzoids, because brady is slow, so they are slowly multiplying. So tachyzoids are fast multiplying and bradyzoids are slow multiplying. Why? Because they will live inside the tissues, inside the cyst wall, and then they will cause chronic infections in the patient. They will live on for years inside the muscles, inside the brain, in whichever organ that they go on and reside. We will look at the life cycle. And the third stage, which is also the infective stage, is the oocyst. This oocyst stage is found in the feces of the definitive host. We will know what the definitive host is. Just remember the cat we spoke about. So you can see that these are the sporulated oocysts. What do we mean by sporulated oocysts? That the oocyst stage has sporozoites inside. So you can see this diagram where this is the oocyst wall, and inside the oocyst you have a sporocyst, right? The sporocyst contains the sporozoites. So this oocyst containing the sporozoites is called as the sporulated oocyst. So now your sporulated oocyst, tachyzoid, bradyzoid, all three stages are infective to man. Now let's look at the transmission here. Okay, so the definitive host of this guy is not man. The definitive host is, what did I say? Let's remember our cat. Our cat. So the definitive host here is feline or cat. We remember what is a definitive host? The host in which the adult undergoes the sexual stages of the life cycle. Okay, so the adult undergoes sexual reproduction, that is called as a definitive host. In this case it is the cat. And what is the intermediate host? It is all mammals, and in our case it is the human, which is the definitive host in this parasite. Now with this, it brings me to an important MCQ and an important thing to remember, which is also very important Viva question. In which all organisms or parasites is man the intermediate host? Okay, so the parasites which undergo a complex life cycle wherein they have more than one host, one is a definitive, one is an intermediate host. So in most parasites, man is the definitive host, but in few parasites, man is the intermediate host. So he's intermediate host in, let's remember with the mnemonic, best plan C. So you know in life we have a plan A, plan B, plan C. So which is the best plan? The plan C is the best plan. Why? Because it helps us remember the mnemonic, right? So how I want you to go about this mnemonic is, I want you to go backwards. So from C we will remember cysticercosis. Cysticercosis. And cysticercosis is caused by, let's jog our memory, Taenia solium. So Taenia solium, when it causes cysticercosis, remember neurocysticercosis, it is only then that the man is the intermediate host, not in case of intestinal teniasis. Okay, now let's go back to the plan. What is the pla of the plan? Plasmodium species. So Plasmodium, which causes malaria, man is the intermediate host. Now let's look at the T. Of course, we are reading Toxoplasmosis. So Toxoplasma gondii is the T. Now I want you to remember, Toxoplasma definitive host is a cat. Cat के साथ आपको क्या याद रखना है? Dog. Cat and dog. Dog. Dog may definitive host, which is the organism which causes infections in dogs. It is Echinococcus granulosus, so that you don't get confused between other parasites with the letter E. So please remember, from Toxoplasma remember cat, from cat remember dog, and from dog remember Echinococcus granulosus. Now S is remaining from our best plan C. S is remaining. We have seen cat, we have seen dog. Now let's look at both cats and dogs. The protestant parasite which is transmitted by both cats and dogs. It is Sarcocystis lindemani. Okay, both cats and dogs. Now B. We remember Plasmodium. Who's the best friend of Plasmodium? Organism with similar disease? Babesiosis. So Babesia species. We know Babesia is best friend of Plasmodium. That is how we remember. So this is best plan C, wherein man is the intermediate host. Now let's go back to our topic, which is toxoplasmosis. We have looked at the transmission is definitive host, intermediate host. We have seen that all the three stages are infective to man. Now how do these stages infect man? So the mode of transmission is two main modes of transmission. One is ingestion and another is through blood, right? So we will see how through ingestion and blood. So blood can be either blood transfusion, it can be organ transfusion, and also placentally from mother to child. This can go. Remember, first thing we said, toxic to pregnant females. So it is going to give congenital infections by crossing the placenta. Okay, so two main modes of infection, ingestion and through blood, either blood transfusion, organ transfusion, or through placenta. Let's look at the life cycle to understand the transmission. Okay, so this is the life cycle. Let's go from point one. Point one, you have the cat. So, okay, I want you to look at two things here. Please see that it is saying infective stage and diagnostic stage. Please find the I's and D's. So, this is the infective stage, this is the infective stage, and this is the infective stage. And which are the diagnostic stages? These two are the diagnostic stages. This is also an infective stage. So, you can see how is it infecting the man? One is blood transfusion, infective. Other is feces, infective. Other is undercooked or raw meat is the infective stage. Now here you can see this is also this is cat litter, right? So this is the feces. So this becomes feco-oral. So you have three routes: feco-oral, raw meat, ingestion, and blood. So you can see the three infective stages in this. Now let's look at the diagnostic stages. What are the diagnostic stages? All three are the infective stages. All three can be found. So all three, tachyzoid, bradyzoid, oocyst, all become the diagnostic stages. Okay. Now let's look at the life cycle. We have the cat. The cat has passed sporulated oocyst in the feces. The sporulated oocyst is taken up by small rodents, forms tissue cysts. When these small rodents are eaten by the cat becomes infected again and passes the sporulated oocyst again. That is how the cycle is maintained within the cats. Now, if this through the cat litter, this sporulated oocyst is ingested by man, he gets infected. So, what is the infective stage at this point? It is the sporulated oocyst. Cat litter infected feco-oral route. Infective stages, sporulated oocyst. Now, if this cat litter is infected, vegetation is eaten by, let's say, pigs and sheep and cattle, and their meat is, if not properly cooked, so raw or undercooked meat of these cattle, when ingested by man, he gets infection. Now, what will be this infective stage at this point? It will not be the oocyst, because it is not, man is not directly eating contaminated food and vegetables. He's consuming the meat, undercooked meat of the animals who have eaten, who gotten contaminated feces. So where will it go? Every time the sporulated oocyst is ingested by anybody, be it man, be it small animals, be it pigs, or anybody, this oocysts will sporulate. The sporocytes will be released inside the intestine of that organism. They will invade the tissues, and the toxoplasma will go and sit in various muscles and organs. Okay, so suppose this pig has ingested a vegetation which was contaminated by cat feces. Now the sporulated oocyst will go inside the pig, will invade the intestine of the pig and go and sit in the skeletal muscles or different organs of this pig. Now when this pig is eaten by man and the meat was undercooked or raw cooked, the infective stage will be taken up from the muscle of the pig. So what is the infective stage which resides in the muscles or the tissues? You remember? Bradyzoid. So in this case, your bradyzoids or the tissue cyst becomes the infective stage. So when I say ingestion through, so through ingestion either by fecal-oral route or undercooked meat, two stages are infective to man: oocyst and tissue cyst. Okay. Now the third is blood transfusion. An infected human who has the organism in its blood, if that blood is transfused to a normal person, or organ transplantation is done into a normal person, or if the mother is infected through the blood by crossing the placenta, the organism can reach the fetus. In all of these cases, infection can occur. Which is the stage which is found in blood? Let's remember tachyzoids, because it is multiplying. Multiplication happens in the blood, inside the lymph nodes, inside the fluids. Okay, so the tachyzoid stage is the infective stage in case of blood transfusion, right? So easy to remember: oocyst through feces, fecal-oral; tissue cyst through meat; and tachyzoids through blood transfusion. Okay, and also placentally. So we have seen human oocyst ingested, okay, goes to the intestine, invades the bloodstream or the lymphatics, goes to different parts of the body. Now, depending on where it goes, if it goes into the eye, it causes ocular toxoplasmosis, simple to remember. If it goes to the brain, it can cause toxoplasma encephalitis or CNS toxoplasmosis. Through the placenta, if it goes to the fetus, it can cause congenital toxoplasmosis, right? And through muscles or any organ, anywhere in the body, it can sit and form the tissue cyst. This brings us to our clinical features. Okay, let's remember our cat again. So how did the cat help us up till now? Cat is the definitive host, feline. We have two more things. Why did the cat have a big head, a spotted head, and red eyes? Okay, and what was the role of CAT scan? CAT scan we will look later. Clinical features: the big head stands for hydrocephalus. Okay, the spotted head stands for intracranial calcifications, and the red eyes stand for chorioretinitis. So I can see that she has three symptoms, right? It's a small cat, it's a baby cat. Hydrocephalus, intracranial calcifications, and chorioretinitis. It forms a type of a triad. This triad is the triad of congenital toxoplasmosis. Okay, congenital toxoplasmosis. The fetus will present with hydrocephalus, intracranial calcifications, and chorioretinitis. That is why fundoscopy or fundal examination is very important in babies. Like you can see here in the picture, you can see ocular toxoplasmosis or chorioretinitis. So this picture can be given to you in your exams. So you should be able to identify. Now, what will it do in adults? In adults, the host can either be immunocompetent or can be immunocompromised. right? So in an immunocompetent patient, it will just cause a self-limiting disease with normal symptoms like cough, cold, bone pain, okay, or pain in the tissues or muscles wherever the tissue cyst is sitting. It becomes a problem in the immunocompromised, okay. In the immunocompromised, if it is an HIV positive patient, and especially if the CD4 count is less than 100, in those patients it becomes a very important cause of opportunistic infections, okay. So in life and in your questions, whenever you have an HIV infected individual with CD4 count less than 100, and you can have toxoplasmosis becomes a very important DD in case of a CNS disease or a eye related pathology. So it causes, sorry, it causes a very important opportunistic infection, which is TE, or Toxoplasma encephalitis. Is a very important CNS disease caused in HIV positive individuals. Other can be chorioretinitis, can also be caused, and other diseases can also be caused, okay. So it takes us to our diagnosis. Now diagnosis is always governed or by the clinical features, right? So we see that it can infect any part of the body. It can infect the brain, it can infect the muscles, it can infect the eyes, it can infect the fetus. So that is how it will help in diagnosis. Now CAT has served two purposes, okay. One is the clinical features, one is the definitive host. What was the third purpose of the CAT? We had said CAT scan, CT scan. So CT or MRI is the mainstay of diagnosis in these patients. So in the diagnosis, we can see multiple lesions or multiple ring enhancing lesions in the brain. So whenever you have such a picture, multiple ring enhancing lesions, you think of CNS toxoplasmosis, okay? So CT and MRI becomes your mainstay of diagnosis. Now another is direct microscopy. We have seen if it was in the brain, you can take the CSF, you can take the blood of the patient, you can take the tissue biopsy from different organs wherever you have a suspicion depending on the signs and symptoms. In the CSF and blood, you will see the tachyzoid stage if you do the direct microscopy, okay. And in front the tissue biopsies, you will see the tissue cyst stage on microscopy. So you can use PAS staining, you can use GIMSA staining. So on different types of staining, you can see these different forms. Now let's come to antibody tests. There is something called as torch tests. You remember? Pregnant females undergo torch test, which is very important. So the TO in torch stands for toxoplasma. R is for rubella, C is for CMV, H is for HSV. Okay, so TO is toxoplasma. What do we detect in these tests? We detect IgG antibodies. Okay, so IgG antibodies. We know that IgG antibodies are also present in chronic infections. So how will we know if the patient is acutely or presently infected? We can check for a 4 times rise in titers of IgG antibodies. Okay, so if there is a 4-time rise of IgG on consecutive samples, then we can say that the patient is currently infected. Another way of finding out current infection through antibody, we all know IgM. The IgM shows acute infections. Okay. Now suppose if the patient is IgM negative and IgG positive, then how will we find out? One is rising titers. Also, what can be done is the IgG avidity testing. So we all know that in the avidity testing, we can find out if the IgG antibodies are more recent or past. So if avidity is less, that means it is a recent infection, because we know that the older the IgG antibodies are, the greater bond they form with the antigens. So we can do IgG avidity testing. Other antibodies we can look for are the IgA and IgE antibodies. They are very important for congenital toxoplasmosis. Okay, for the congenital toxoplasmosis. Why? Because IgM, IgG will not cross the placenta. So how will you find out if the fetus or the child has infected? You can check for IgA and IgE antibodies. What is important in IgA is persistence of IgA beyond 10 days of birth. Okay, this is important. So if IgA persists beyond 10 days of birth, then it is a marker. Now let's look at a gold standard test for toxoplasmosis. Gold standard but oblivious, old, we all know. It is called as the Sabin-Feldman dye test. Okay, please remember Sabin-Feldman dye test. What do we do in the Sabin-Feldman dye test? Okay, we take the live tachyzoids. Tachyzoids are the trophozoite stage. We take live tachyzoids, okay, and we put it in two types of serums. One is the serum which has specific antibodies to the tachyzoids. Another is a normal serum. The normal serum will not have the antibodies. Okay, so when we put this serum containing the antibodies on these tachyzoids and I look under the microscope, we will see that the tachyzoids are thin and destroyed. Okay, because of the antibodies. And in the normal serum, the tachyzoids are swollen. Right? What do I do at this point? At this point, I add methylene blue dye to help me visualize better. That is why I'm writing it with the blue pen. Okay, so we know that we have added the methylene blue dye at this point. We can see live tachyzoids, serum with antibody, thin and destroyed, live tachyzoids, normal serum, swollen. If I add methylene blue, what will happen to the thin and destroyed tachyzoids? They will stain. So when I say that less than 50% of the tachyzoids are stained, that means the serum had antibodies and Sabin-Feldman dye test is positive. If 90 to 100% of the tachyzoids are stained and swollen, that means it is negative or the antibodies are not present inside the patient's serum. Okay, so this is the Sabin-Feldman dye test, which is the gold standard but oblivious test. Okay, the fourth method, the another thing similar to this is the immunofluorescent assays. These are the modern technologies. Instead of Sabin-Feldman dye test, we can on a plate coat the antigen, put the patient's serum. If antibodies are present, antigen antibody complex is formed, fluorophores are attached, and we see fluorescence. So like any other normal immunofluorescent assay, these assays can also be done. So what is important to remember, the Sabin-Feldman dye test is an example of complement mediated neutralization test. Okay, it is a complement mediated neutralization test. This is a one word question. Okay, now another thing which can be done is, we have seen what we have seen till now. We have seen direct microscopy, we have seen CT scans, and we have seen antibody tests. Other thing which can be done is molecular tests. Obviously in today's day and age we have to do molecular tests where PCR can be performed. On what all can PCR be performed? It can be performed on body fluids such as CSF or wherever. It can be performed on tissues, tissue biopsy. Can be performed on the amniotic fluid of the pregnant females. Okay, in the amniotic fluid to detect the congenital toxoplasmosis. Okay. Other method is animal inoculation, which is an old method but still used for research purposes. Animal inoculation is done in the mice and it is done intraperitoneally. Okay, intraperitoneal injections in the mice. Other method, if you don't want to use the entire animal, just use the tissues or the cell lines. So tissue or cell line cultures can be done in order to obtain the organism. Okay. Remember, for diagnosis in adults, any of these methods can be used. For the diagnosis of congenital toxoplasmosis, the diagnosis can be done either antenatally or it can be done postnatally. Postnatally, we have seen the IgA, IgE, or the IgM can be detected in the neonates, right? Antenatally, we can do the PCRs in the amniotic fluid, or we can also do the IgM of mother. Okay. So, in neonates, in mothers, you will do the torch test. In neonates, you will do fundoscopic examination and antibody testing. Okay. So, let's come to the treatment. So, if we simply remember our cat, big head, spots, red eyes, cat scan, feline, we can sum up the entire toxoplasma. So, in treatment, the mainstay of treatment is pyrimethamine plus sulfadiazine plus folinic acid. This is the mainstay of treatment of toxoplasmosis. So, in case of an immunocompetent host or an immunocompetent adult, this therapy needs to be given for 4 to 6 weeks, followed by assessment. You assess the patient again and then you see. Okay. In case of an immunosuppressed individual, this is done 4 to 6 weeks beyond the resolution of signs and symptoms. So, the treatment will go on for more than 6 months in these individuals. Okay, 4 to 6 weeks, symptoms resolve, more 4 to 6 weeks. Okay, in case of congenital toxoplasmosis or for neonates, this treatment is given for 1 year. Okay, for 1 year. Another drug is spiramycin. Okay, spiramycin. So spiramycin, it concentrates in the plasma, in the placenta, concentrates in the placenta. So it leads to acute, it can be given in acute toxoplasma infections. Okay, in acute toxoplasma infections, if it is detected in less than 18 weeks of gestation, okay, and the fetal status is not known, in those cases pyramycin can be given. Okay, if it is more than 18 weeks or the fetus status is known, then the standard treatment needs to be followed. In case of sulfa drug allergies, other things which can be used are azithromycin or clindamycin. In patients with lesser CD4 counts, in HIV positive patients, cotrimoxazole is also added as a prophylaxis. Okay. So this finishes our topic, toxoplasmosis. Please remember our cat and we can sum up the entire topic. Thank you.